Okay, so our next topic we have RNA isolation. So review lang natin. So remember the central dogma that all information come from the DNA, and then this DNA, we use DNA as a template to make RNA, and that process we call transcription. And then yung ibang process naman where we use RNA to make DNA, we call it reverse transcription. Okay. So from RNA, we use RNA as a template to make a polypeptide or protein, and we call that thing as translation. So what is the message behind this? So if we wanna quantify protein, like how much protein in the cell, we could use RNA, like we could isolate RNA. But remember, sometimes the level of RNA doesn't reflect protein level. Why? Kasi sometimes we have a lot of mRNA, but those mRNA is not translated into protein. Pwede yun. Another thing, minsan, onti yung mRNA or RNA natin, but the protein level is high. It could be that walang transcription, but we have protein retention in the cell. So pwedeng mangyari din yun na stable yung protein level sa cell. So hindi na kailangan ng transcription to make more protein like that. Okay? So again, pwede tayong mag-speculate na maraming RNA means maraming protein, pero hindi laging ganun nangyayari sa cells. Okay? So another example, we have structural genes. Usually sa bacteria, meron tayong mga structural genes. Kunwari si B, kailangan niya si A at si C at si D, or I mean yung protein A, B, C, and D, magkakasama sila together to form a big protein or complex protein. Okay? So, pag kasama sila sa subset at meron silang isang promoter, sometimes we call it operon. Okay? So, this is the fourth question. So, fourth. So, hanapin niyo kung nasan yung question 1, 2, and 3. So, yung fourth question is yung what is an operon? Okay, itong operon. And what in bacteria, anong function ng operon? Okay. So again, we have the DNA, which contains the instruction. Okay. And then that instruction, kailangan i-transcribe into mRNA. And then that mRNA should be translated into polypeptide or protein. And pwede natin gawin is to do mRNA extraction or total RNA extraction. So usually, sa bacterial identification, we use 16S rRNA sequencing. Therefore, to get the sequence of your rRNA, we need to isolate total RNA ng bacteria. And this is the fifth question. So yung fifth question, why is it we use 16S rRNA for bacterial identification. Bakit hindi yung other gene? Bakit hindi yung ibang gene? Bakit usually 16S rRNA yung ginagamit natin to identify bacteria? Okay? So, pag nakapag-extract tayo ng RNA, again, we could use that RNA for identification such as 16S rRNA. We could use that RNA to quantify or to speculate kung yung protein na yun or yung RNA or yung RNA which will be translated into protein ay nag-increase or nag-decrease dun sa ating cell. Okay? So, ano yung ways natin to isolate RNA? So, of course, we have the source. Could be serum, blood, broth, mice, tissue, or cells. And then we need to homogenize yung ating sample. And we use this guanidinium phenol. Okay? And yung pH niya is low. And important yung pH for this extraction. So usually, after when we add this guanidinium phenol together ng ating sample, so yung RNA, protein, DNA will be dissolved together. And then if we add chloroform, ayun na yung way natin for phase separation. So usually, the RNA goes to this aqueous layer, and then we have intermediate layer here, which contains the DNA, and meron tayong lower layer here, yung organic layer, which contains protein and some DNA. And then after that, after the separation phase, we could collect tong aqueous layer, which contains the RNA, and then add isopropanol for precipitation of RNA pellet. So after isopropanol precipitation, we already have the pellet. We can wash the pellet. So pwede tayo maglagay pa ng another step here. Washing the pellet with ethanol, 70% ethanol. And then after that, remove the ethanol and we air dry the pellet and resuspend the pellet with water. Okay. So, yung RNA extraction is simple lang, and we use that RNA for cDNA synthesis na pwede natin gamitin for PCR reaction. Or sometimes, yeah, we could make PCR reaction, and then after that, those PCR products we could use for sequencing and comparison for bacterial identification. Okay, so watch natin kung paano tayo mag-isolate ng RNA using trisol reagent. So yung trisol reagent usually is made up of guanidinium phenol.
Hi guys! So today, nag-isolate tayo ng RNA from blood serum. Okay? So medyo maingay yung machine. So sana naririnig nyo ko. So gagamit tayo ng trisol reagent. So later explain ko anong purpose ng trisol reagent for RNA isolation. So kuha lang ako ng tubes and labelan ko yung tubes and I'll show you how to isolate RNA from mice blood serum. Okay?
Okay. So again, gumamit tayo ng trisol, which is actually made of guanidinium phenol. And what is good about trisol reagent ay inactivate niya yung mga RNAs. So yung RNAs, those are enzymes that could degrade RNA. So we need to inactivate those enzymes. And another thing is yung mga proteins in the cell or in our sample, usually, dinid nature din no ating trisol reagent. So, nag-prepare lang ako ng tubes for our experiment. And then after labeling, tandaan nyo, dapat laging may label.
So first thing, ito yung serum natin at kukuha tayo ng 100 microliters of serum and then transfer natin dito sa clean uh, epi tubes. Okay? So,
So, ayan, naglalagay lang ako ng 100 microliter of serum. So after that,
okay, so we're finished transferring here, our samples. So now we need to add 1 ml of our trisol reagent.
so make sure na madami yung trisol reagent.
We need to vortex the sample. And then we need to spin down. So we need to spin down the sample to make sure na walang may iwan dito sa taas ng tube. Okay. Sa cover ng tube. Okay, so we're done adding the trisol. So ganito yung itsura niya. And after that, we need to put 200 microliters chloroform. Okay? 200 microliters. So after chloroform, we need to vortex the sample like for a few seconds. And mag-wait tayo ng 3 minutes para mag-form ng layers. So, ganito yung itsura muna niya pag may chloroform na siya. Okay? So, as you can see, may layers because of different polarity of trisol and chloroform. Or vortex. Okay. Okay.
So naglagay lang ako ng chloroform.
Okay. After 3 minutes, ganito na yung itsura ng sample natin. And then we need to centrifuge this for 12,000 G for 15 minutes. Okay? So punta na tayo ng ating centrifuge. So ito yung aming centrifuge ng Jurassic Park. At 12,000 G or in RPM. 12,000 G. Legal. Welcome to Jurassic Park. Okay. Lalagay na natin sample. Lalagay na natin. Okay, so tapos na tayo mag-centrifuge. Kung mapapansin nyo, merong pink layer, tapos merong white dito, parang white, and then merong, sorry, a white, and then merong clear layer. So tatlo yung layers natin. Itong pink, then meron kayo makitang parang white dito na film, and of course, meron kayong clear layer. Oh my God! Ah! Ah!
So, class, so tandaan nyo, yung pink layer, ayun yung organic part. It contains a lot of protein. And then yung middle layer, intermediate layer, it contains the DNA. And the aqueous part, yung upper layer, it contains the RNA. That's why medyo frustrated ako na nalaglag yung tube. Dahil na-mix ulit yung DNA with the RNA layer.
So, pag nalaglag siya, kailangan nyo lang i-recentrifuge ulit. Okay?
So, yun yung idea doon. Kapag na-mix ulit yung sample, you just need to centrifuge again. Hi.
Okay, so ang gagawin lang natin ay kunin yung clear layer and then transfer natin sa another test tube. So make sure na carefully nyo i-transfer yung clear layer dun sa other test tube. So, yun yung ating technique na kailangan. Okay, yun yun.
So, kapag gagawin nyo siya, make sure na wag yung isama yung outer layer. So, wag nyo na lang kunin lahat. Ang ginagawa ko, nagtitira pa ako ng konti dun sa tube para hindi mahalo yung aqueous layer dun sa intermediate and organic layer. So, ayun. Label lang and then get all the aqueous layer.
So, na-transfer na natin yung upper layer. And then after that, we need to add ice cold isopropanol or isopropyl alcohol. And after adding isopropyl alcohol, we need to vortex it. And then after ma-vortex, put lang natin set aside for 10 minutes. Okay? And then after 10 minutes, we need to centrifuge again the samples, like 12,000 G for 15 minutes. So, ganito yung reaction niya with alcohol. So, nag-add lang ako ng 500 microliters of isopropyl alcohol. Okay. So, pansin nyo, halos wala namang changes dun sa ating sample. So,
So, kailangan tayo ng alcohol. Remember, yung isopropyl alcohol is important to precipitate our RNA.
okay, so nakuha na natin yung sample out of the centrifuge. So, ganito yung itsura niya. So, pag marami kayong RNA, usually may makikita kayong white pellet here. Pag wala, so minsan super onti lang ng RNA na na-isolate natin. So, after this, pour out lang natin tong chloroform.
So, pour
Okay.
out yung
So again,
chloroform.
after this, we just need to pour out the chloroform. Okay, so
So, pour out lang yung chloroform, isopropyl, sorry, not chloroform. Pour out lang natin yung isopropyl. Correction. We need to pour out the isopropyl. Okay?
so after nating ma-pour out yung ethanol, maglalagay na tayo nitong 75% ice cold ethanol.
I think, guys, I'm very tired na. So, pour out the isopropyl, okay? Correction. Pour out the isopropyl, and after we pour out the isopropyl, we add the ethanol, okay? So, yung ethanol yung ating gagamitin to wash some impurities in our RNA.
Okay? And then centrifuge ulit. Okay, so tapan na natin yung chloroform part. So this time we need to add 75% ethanol.
Okay, again, class, yung itinapon kanina ay isopropyl, and then after we remove isopropyl, we just need to add ethanol. And the ethanol, we just use it to wash away some impurities, okay? So I just add ethanol.
Okay, so we're done centrifuging the sample. Then we just need to pour out everything. Then for drying, usually nilalagay ko lang siya ganito sa tissue paper.
Okay, so we just need to pour out the ethanol, okay? And then, wala kang masyadong makitang pellet sa ilalim, pero may pellet yan, okay? So, ibig sabihin, kapag konti yung RNA, ang pellet sometimes not visible, but it's there, okay? So, wag kayong mag-worry kung wala kayong nakikita. But believe me, there is RNA at dito sa bottom of our epitubes.
To remove yung excess ethanol and air dry for 5 to 10 minutes. Then we put our RNAs, DNAs free water to dissolve the RNA. Okay.
So that's the end of our experiment. And don't forget to submit your homework.